to introduce today's speaker, Dr. Neil Benson. Neil is the head of Quantitative Systems Pharmacology Operations at Certara. He has extensive experience of using modeling and simulation to address questions of critical importance in drug discovery. Prior to coming to Certara, he was a founder of Xenologics, a consultancy focused on applying PK/PD modeling and systems pharmacology to drug discovery. Xenologics became part of the Certara family in 2015. Neil was educated at the University of East Anglia in the United Kingdom and the University of Lund in Sweden. He earned a first-class honors degree and a PhD in enzymology. Neil, welcome to today's event. I'll now turn it over to him to begin the presentation.
Okay, thank you very much, Suzanne. Afternoon, everybody. Can you go back one slide, please, Suzanne? So we should be on slide 1. So today I'm going to talk about the application of systems pharmacology, which is a mathematical modeling approach to integrate our understanding of biology and drug pharmacology and PK. And I'm going to talk about the application of that to a very topical and interesting area, which is the clinical development of inhibitors of an enzyme called fatty acid amide hydrolase that have been looked at for pain, amongst other diseases. And as Suzanne said, my name is Neil Benson. I'm the head of QSP operations at Certara. However, I should acknowledge that much of the work that I talked about was done during my time as an employee of Pfizer. Next slide, please. So what I'm going to do today is give a summary overview of the work that was done in this area, but there is a more detailed description of the work in 2 different publications. One, a kind of more summary level description in CPT, and then the second, a quite detailed publication that describes the models and some more of the background. So if you're interested in finding out more, please look at these publications, and also you have my contact details if required. Next slide, please. So I'll just start with a little bit of a story, if that's okay, because I think it's relevant to the picture here. So this is something that I recall from being a small child, something I learned, a very important lesson. And I was once walking with my young friends and buddies in the forest, and we came across a wasp's nest. And one of my friends said to me, why don't you go over there and poke that wasp nest with a stick, because that will prove to us how brave you are. And I remember thinking at the time, it would be a good thing to prove to my friends that I was brave. But on the other hand, part of my brain was telling me this isn't a good idea because this could be dangerous. But unfortunately for me, I allowed my friend to persuade me that it was a good idea. We looked at the wasp nest. He made the point, it doesn't look very big. There's only a few wasps coming and going. How dangerous can it be? But unfortunately for me, I believed this, and off I went, and I gave the nest a poke with a stick. And at first, nothing much happened. So I poked it again, and then, of course, there was a loud buzzing noise, and lots of very angry wasps came flying out, and I had to make a run for it, and I was stung for my trouble. And the lesson that I learned there was manifold. One of them was don't necessarily believe what everyone says to you, and also that sometimes it's important, no matter how benign something looks, to look into it carefully before you kind of move ahead. And Shakespeare picked up on this very nicely in Henry IV, and this sort of famous quote came from that discretion is often the better part of valor, or sometimes, or put another way, if you don't take appropriate precautions, you can sometimes be stung. So can you move to slide 4, please, Suzanne? Thank you. Before we move into the science of the presentation, I just wanted to make a couple of points more generally about why we should be interested in this new approach, this systems pharmacology approach to drug discovery, not just specific to pharma, but in general terms. So one of the main reasons, I think, is because it's one way of trying to tackle the problem that we have with attrition and return on investment. So any observer of the productivity in pharmaceutical research will know that by all metrics the productivity is declining. And next slide, please, Suzanne. Slide 5. Yep. This has been looked at by numerous groups over the years. So in Kohler and Landis in 2004, Paul and his colleagues in 2010, and latterly 2014, Hay et al. Looked at this as well. And broadly, they all came to the same sort of conclusions that we see a big problem in attrition. In other words, drug programs failing, and they fail in, most importantly, in the phase 2 part of drug discovery. And the other conclusion that I would draw is that nothing really has changed since 2004. The 4, 10 and 14 papers all quote similar statistics. Next slide, please, Suzanne. So what is the problem? I believe if you look at why we see this attrition in phase 2, mostly it's to do with one of two things. Either the drug didn't work, it didn't do what we wanted it to do, it had no efficacy, the doses we tested, or it exhibited some kind of unexpected toxicity. And I think if you dig into why that is, there's a clear explanation, and that is that we didn't fully understand the biology. We didn't it's a very complex system, and we didn't understand the consequences of perturbing that complex system with a drug, and which leads to unexpected effects. And this is commonly observed. So we're often in the position like the two chaps in the middle of the figure here, where we have a lot of input data. We have some data around the emerging properties, but when it comes to understanding how the two things connect, we're limited. Now, this is not a unique problem to drug discovery or biology. This is a problem that's observed in other areas, such as engineering and physics and so on. And they've been very successfully employing the use of the kind of mathematical modeling and simulation approaches that I'm going to talk about today in those areas. And so I believe that we can do the same in biology. The next slide, please, Suzanne. So now we'll talk about an example of that, and as I said, that's applying this kind of mathematical systems pharmacology model to understand and think about the pros and cons of FAAH, fatty acid amide hydrolase, as a target. So for those of you that don't know a little bit of background biology, so this is a very, very interesting and hot topic. It's been understood for a number of years that there are receptors for cannabinoids, CB1 and CB2, in the body, and CB1 is thought to primarily be present in the brain, and CB2 in the periphery, and that these are the target for the active ingredient of cannabis, which is tetrahydrocannabinol. And also that there's an endogenous cannabinoid called anandamide, which is, or AEA, which is also an agonist of CB1 and CB2. And there's been a wealth of interesting biological data generated around this in the last decade or so. For example, showing that increased levels of anandamide has an effect such as attenuating nociceptor firing, amongst other things. And certainly some would claim that there is evidence of efficacy of cannabis preparations in pain, so potentially some clinical precedent here. Next slide, please. So it's a very compelling story. So just to look at the basics here, as I said, anandamide is believed to be an agonist of CB1, which has numerous downstream effects which culminate in potentially the attenuation of nerve firing. And anandamide is degraded by the enzyme FAAH. So the project hypothesis here was that there's agonist drive through CB1 via anandamide and via to arachidonoyl glycerate, or 2AG, and by inhibiting FAAH we will increase the local concentrations of anandamide, and in turn that will decrease nerve firing and attenuate pain. And we were able to, or Pfizer was able to generate a molecule, 445-7845, that was a very potent inhibitor of the FAAH enzyme, and also that this was positive in certain animal models of pain. So the next slide, please. So the molecule was tested in 11 rodent models of pain. The green represents those where the molecule was positive and the red where it was negative. So there were a number of positive results as well as a number of negative ones. And the project's interpretation of this data was that the FAAH inhibitor is likely to address specific pain types that are represented by the model. So each of these models represents a specific model of pain for a specific type of pain, such as chronic pain or acute pain, and therefore the molecule could address these different pain states. Next slide, please. So, a very compelling story, interesting biology. We had good compounds, and we had a biomarker which was anandamide that we believed we could measure in preclinical species and patients. But immediately prior to the phase 1 evaluation of this molecule, the clinical pharmacology team came to me with the following questions. So one of them was, will we test the pharmacology fully at the doses that we're intending to use in the patient, the volunteers, and patients ultimately, and can we predict the receptor occupancy that we will see? And can we also predict what level of occupancy would be required to get the efficacy that we want? And that's a very complex question. And so we used the systems pharmacology approach with internal and external data. And as I said, this work was done as 7845 approached phase 1, so in the preclinical part of drug discovery. Next slide, please, Suzanne. So the first stage in all systems pharmacology projects is to do a wide sweep of the available literature to find useful facts, parameters, and information for the model building exercise. And we found out a number of things in this. So the first one was that there are some very useful data. So I'll show you this in a bit more detail in a moment, but there are good data on the disposition of anandamide, good data on the enzyme kinetics of the biosynthesis and degradation of anandamide, so information that would allow you to build a model of the disposition and anandamide, essentially. So that was pretty rich, actually. However, there was a lot of information on the endocannabinoid biology as well, because what I've shown you before is a sort of top-level summary, but there were lots of kind of open questions in our view. So, for example, that anandamide reportedly an agonist of CB1, which in principle would have a pain attenuating effect, but also being an agonist of TRPV1, which would be more commonly associated with causing pain rather than kind of inhibiting it. And so, yeah, there were lots of questions about the steady state impact of perturbing anandamide. The third thing that came out was, and this was in my sort of initial simple diagram, was that 2AG and anandamide both agonize CB1 in principle, but to our knowledge, there wasn't a great understanding of how those two things would interact with one another. And then finally, the link between occupancy and the firing of nociceptors, although there seemed to be some qualitative evidence for that, we couldn't find any quantitative data that described how occupancy would relate to a decrease in firing. And so, therefore, it would be difficult to specify what success would look like in terms of receptor occupancy versus time in the patients. The next slide, please. So we, taking that information, we constructed a model. So it was comprised of the 5 components you see here. So the first was a model of disposition. So by XEA we mean not just anandamide, but there are other ethanolamides that could be important in the system here. And so the model was built to include those other ethanolamides and describe the disposition of those ethanolamides in terms of their tissue binding and compartmental localization. The second element of the model was related to how the FAAH would bind anandamide and these other ethanolamides because it's a multi-substrate enzyme, and obviously that could influence our interpretation of what was occurring here, and so that was integrated in the model. A model of the biosynthesis and degradation of anandamide based on the literature. Fourthly, the clinical PK and pharmacology of the molecule, and then finally a model of how the anandamide interacts with its receptor. And all of that was integrated together into an ordinary differential equation model that we believe described the biology. Next slide, please, Suzanne. So, looking at the top left-hand corner of the slide, we did some simulations. So we were testing our thinking by using the model to simulate what would happen if we gave the doses that we were expecting to give in the clinic. And so we predicted a couple of things. So the first, and that's shown in the top left-hand panel of this figure. So the first thing we predicted was that the anandamide levels in the plasma of the volunteers would rise, as shown in the blue line. It would reach a peak, and then it would decrease away depending on the dose given. So that was good news from the project's perspective, because we were predicting a measurable effect in the plasma of the patients. However, and as is shown in the bottom left-hand corner of the figure here, when we took the drug to man, to the volunteers, we saw something slightly different. We saw an increase in the level of anandamide in the plasma after the patients were given drug relative to baseline, but that seemed to reach a plateau that decayed back to the baseline eventually, and it seemed also that plateau was independent of dose. And so the first thing we concluded there was that our model was wrong. And now, one interpretation of that is that's a problem. However, I take the view that sometimes models are much more useful when they're wrong than when they're right, because they allow you to ask some questions. And so in this case, we asked ourselves the question, well, our model's predicting a very sharp peak and decline. We actually see a plateau. What is it that's missing from the model that would allow us to explain that plateau? And the conclusion that we came to was that something else must be clearing the anandamide other than FAAH. And so that if you include something in the model that clears anandamide in addition to FAAH, then you can observe this kind of plateau that we saw in the initial data. Next slide, please. That led us to the question, well, what are the potential candidates? And the answer to that was a huge number of things. So, for example, cytochromes P450 could potentially metabolize anandamide, lipoxygenases, cyclooxygenase, and so on. So there was a long list of potential candidates of enzymes and other routes that could actually clear anandamide in addition to FAAH. Now, one of the values of the model, as well as allowing us to kind of ask that question, was that it's a quantitative framework. So it has a quantitative dimension that allowed us to ask the question not just which processes could be clearing the anandamide, but what are the properties of that process? What are the kinetic properties of it? And so we would be able to match those with the known properties of the different clearance processes that we had in our list, and ask the question which one fits best with the model. So we use it as a way of triaging this sort of long list of potential candidates. And we concluded from that exercise that the most likely candidate was an enzyme called N-acyl ethanolamine hydrolyzing acid amidase, or Na cubed. And so I think this is an example of the added value of systems pharmacology, not just to identify what a missing process could be in general terms, but very specifically generate some hypotheses about what it might be. And I'll come to why this could be important in a moment. But next slide, please, Suzanne. So now we've integrated this additional clearance process into the model, we were able to simulate what we thought the effect would be and compare that with the actual data that we saw in the patients. And that's what's shown in the 4 panels on this slide. So you can see, and so the solid lines are the simulations of the model, and the kind of solid blocks in different colors are the actual data. So you can see from this slide that we were able to simulate very accurately the effect that we actually saw in the patients for multiple endpoints. So not just for anandamide, but also for some of the other ethanolamides that we included in the model, and that we could describe the time course and dose response of those very, very accurately. So we have a model that describes the biology as we know it very precisely. Next slide, please. So the first question, however, that came back from the project team as well, we can see we're increasing anandamide in the plasma. We're reaching a maximum of about 10 nanomolar in terms of the anandamide, but we also know that anandamide binds very strongly to human serum albumin, so the free fraction is sort of the order of about 0.1%, so it's 99% bound to human serum albumin. And also that from our in vitro experiments, we were determining a primary pharmacology or an EC50 in our kind of in vitro assays in the region of 300 nanomolar or thereabouts. So given this free fraction, given this number, given the peak anandamide of 10 nanomolar, and making the typical free drug hypothesis assumption that it's free drug in the aqueous phase that's driving effect, how can there be an effect? Because the peak-free anandamide is peak molar, and if you work that through, given these assumptions here, these values here, that would translate into significantly less than 1% occupancy. So that wouldn't seem to make sense even for an agonist. So the next slide, please, Suzanne. Thank you. So there may be an explanation for that. And that's summarized on this slide. So there is a school of thought that anandamide is atypical in a way that it does not concord with the sort of standard free drug hypothesis, in that it, rather than binding the receptor from the aqueous phase, facing phase, rather it binds or intercalates within the membrane first, and then it diffuses to the receptor target CV1, and then it expresses its pharmacology. Now, this we think would explain why we might see some pharmacology at these levels of anandamide if anandamide is a very hydrophobic molecule, because the concentration of at steady state of the anandamide would be much higher within this kind of environment, the lipid environment, than it would be within the hydrophobic environment. And this is in agreement with observed and calculated data for anandamide, because it has a very high log D in the order of about 6. So you would expect it to be much more, maybe 6 orders of magnitude more concentrated in the hydrophobic environment than in the hydrophilic environment at steady state. And you can express that mathematically using the equation that's shown in the middle of the slide here, whereby the kind of observed KD in this instance you see is dependent on the binding of the drug to the receptor, or anandamide to the receptor, multiplied by its partition coefficient, which is in this case is a very small number because of the high log D. And so we think this kind of hypothesis can explain why these free levels, apparently very low free levels of anandamide, can actually drive some meaningful pharmacology. Next slide, please, Suzanne. So we have a model that we think describes the biology as we understand it. We can show that it can predict observed clinical behavior. And so now we use the model to try and answer the original question of the project, which was, well, at the doses we're going to test, will we inhibit the target, and what will that mean in terms of receptor occupancy? So the answer to the first question was, we thought that we would inhibit the pharmacology as much as possible, giving us each of the doses here giving us in excess of 96% FAAH inhibition over extended periods. So significant pharmacology at the target for sure. However, we're predicting that receptor occupancy would reach a maximum of about 25%, and that would be independent of dose. So it doesn't matter how much extra inhibitor we give, we can never increase the occupancy above that level. We can only extend the time course of it. And because the occupancy is limited by the clearance of anandamide by something else, which we inhibit, we hypothesize is Na cubed. And so the question there is, well, is 25% occupancy of the receptor enough to give a pharmacologically relevant effect? Next slide, please, Suzanne. So just to recap where we've got to. So the kind of what I would call the traditional drug discovery view here was that there's a very clear rationale, so there's a very compelling and interesting biology. There's apparently some clinical precedent for using cannabinoids in pain. We had very good biomarkers that we could measure preclinically and clinically in a very robust way and show a clear PK/PD response. And we had animal model positive data. So this, for the US audience, a slam dunk. For those who are more into the soccer, it's a Messi through on your goalkeeper one-on-one. So this looked like very, very positive, I think, was the perspective. Next slide, please, Suzanne. However, the systems pharmacology perspective is quite different. So first of all, I think although there is a hypothesis about why we might expect to see some meaningful occupancy, there are some questions about that. There are questions about the receptor occupancy levels that we can achieve, and is that enough? Is 25% enough? And also, actually, we don't have control of it because it's limited by something else that's clearing the anandamide, probably. We don't understand how to relate occupancy to nociceptor firing. So, is 25% enough to decrease the nerve firing? Maybe not, but we don't know, essentially. And then, do we know how much nociceptor firing would need to be decreased to have an impact in pain? No, so we didn't understand that either. There's no quantitative data on that. And then finally, the endocannabinoid system is very complex. We looked at a kind of slice of it, as it were, the anandamide CB1 axis, but we're not looking at the other elements, such as the 2AG, and for example. And so the question there is, well, what is the impact of looking at a kind of one element of the system and ignoring the others? And so, all in all, we concluded that this actually is a relatively high-risk target. The next slide, please, Suzanne. So the feedback that I gave to the team prior to the phase 2 clinical trial in 2010 was that the model suggested that the doses planned, we would test the pharmacology as far as possible, almost completely inhibiting the FAAH enzyme, but that there's an alternative clearance pathway for anandamide, which means that the effect of any FAAH inhibitor would be limited. We hypothesize at this point in time that it's this enzyme Na cubed. There are no quantitative data that would link occupancy to the pain outcome. So the success criteria is an open question. We predict from the model some occupancy, but is it enough? We don't know. And then that led us to the other point, which was made by Paul Morgan and his colleagues in a paper in Drug Discovery Today in 2012, which was that if you don't have a way of showing that your pharmacophore gets to the target and expresses the pharmacology, you run the risk of running an experiment that you can't interpret, because if you, in the event of a negative, you won't know whether it was a negative because the drug didn't get to the target. In this case, I would argue that's anandamide, and express the pharmacology in the way that you wanted, or whether the mechanism, the hypothesis was void anyway. And you can't discriminate those two things without having an assay of pharmacology. And so ultimately this is a high risk project, and it's a high risk that we run an experiment we can't interpret. Next slide, please, Suzanne. So there is an epilogue here. So 4457A45 was taken to the clinic in 2012 for a phase 2 study in OA patients, and it failed. It didn't do anything in terms of the drug, in terms of the end point in OA. So clearly lots of questions questions about the confidence in our animal model translation. So we saw some apparently very clear positives in animal models, but they haven't translated to the clinic. The question about did we express the pharmacology at this point in time, we don't know. And so the systems pharmacology perspective at the time was, well, we really need to build our knowledge and technology here before there's any further work in this area. Next slide, please, Suzanne. And we commented on this in the CPTPS paper in 2011, where we said that any future progression of FAAH inhibitors should be accompanied by efforts to develop technologies that will enable demonstration of pharmacology in CB1, but also that we really need a better quantitative understanding of the relationship between occupancy and attenuation of any nociceptor firing, together with an improved knowledge of how the components of the endocannabinoid system behave in humans, because to us it seemed like this is a potentially very powerful pharmacology, and so a kind of integrated system knowledge would be extremely valuable. Next slide, please. So there are further epilogues here, so not to pick on anyone in particular, but other companies subsequently looked at FAAH as a target. Vanalis, for example, and as for Pfizer, it failed in pain, didn't have the required efficacy. The next slide, please, Suzanne. And unfortunately, as probably many of you know, a company called Bial took forward a FAAH inhibitor at the beginning of this year, end of last year, and there were some terrible events. Unfortunately, one person in the volunteer group died. 6 patients were hospitalized. So it was a very, very sad and tragic event. Next slide, please, Suzanne. So the French authorities put together a panel who published a document reviewing what happened in that clinical trial, and that was published just last month. My view, the conclusions were that the molecule itself was probably the main candidate for the toxicity, that it was dosed to much higher concentrations than would saturate FAAH inhibition, which the authorities kind of highlighted. But for me, more importantly, I don't think ultimately we still don't really understand what the origin of the toxicity is. And to me, that again points to this question of, well, did we really understand the complexity of the biology enough to be able to understand what the consequence of perturbing it would be, good or bad? Next slide, please. And so how do we move forward from here? So I think this is clearly a very interesting area, and there could be some useful drugs that could be developed by looking at this. However, I think we do need to understand it better than we do at the moment. So one thing I think immediately is that, as I said in the beginning, we kind of simplified our model of the system, as it were, to the anandamide facet of the biology. But we know already that there could be an important element, which is agonism driven through to AG. Now, obviously, it could be that the biomolecule is toxic because it hit something that we don't know about. That's always possible. However, it seems to me that if we are going to look at this, the most logical next step would be to start asking questions about, well, which enzyme is or which protein or which receptor is the molecule that's been designed most likely to hit. And it seems to me, because it's been designed to inhibit FAAH, then looking at closely related hydrolases would be an interesting, probably the most interesting place to make a start. And so that would include enzymes like maglipase, and then there are recently been discovered these alpha beta hydrolase domain 6 and 12 enzymes that are thought to be could be important in turning over to AG. So the next slide, please, Suzanne. So I could imagine that with that, under the assumption that there is some interesting data there, that we could start to build and develop our knowledge of the biology to include the parts of the endocannabinoid biology that we know are there, but are not currently kind of factored into our thinking. So on the one hand, anandamide binding and agonizing CB1, but also maybe there's a role for 2AG, and maybe maybe there's something here around, or does Bial interfere with any of these processes and change the steady state behavior of 2AG, for example? And so then we can start to build an extension to the existing systems pharmacology. However, this is a very complex question, so I think it requires a mathematical systems pharmacology approach. Next slide, please, Suzanne. So just to draw some conclusions, I think one of the main added values of the systems pharmacology approach here was to give a perspective, a different perspective to the traditional drug discovery approach. So I think we saw very compelling biology. We saw apparently very compelling positives in animal models, and so that all looks very good from a traditional drug discovery perspective. But the systems pharmacology perspective, where we had to ask quantitative questions, gave a very different one. And I think that allowed us to highlight a risk of failure, and it suggested testable hypotheses. So one of them is that we think that this Na-cubed enzyme could be important in clearing anandamide. So that could be tested if necessary, I think. And it also highlighted that even relatively speaking, our understanding of the biology here was very limited. So there are lots of complexity in the endocannabinoid biology, lots of negative feedbacks, and those sorts of things are very difficult to predict without some kind of system model-based understanding. We don't know about the role of 2AG and whether changes in this, how that would interact with changes in anandamide. As I said before, we don't really understand the receptor occupancy and nociceptor firing relationship, and so the success and safety criteria here are unknown. And I think that the events are so complex that it would be very difficult to move forward without some kind of model-based mathematical understanding. Next slide, please, Suzanne. So I will stop there. Thank you very much for taking the time to come to our seminar this afternoon, and I will take any comments or questions, I believe.
Okay, thanks so much, Neil. As Neil mentioned, we're now moving into the Q&A portion of our webinar. We'd like to encourage our audience to submit your questions to Neil through the Q&A box, and please select all panelists when you submit your questions. So Neil, it looks like we have a question from our audience. It's easy to be smart in retrospect. What is the value of systems pharmacology prospectively? Okay, so can you just go down a couple of slides to the backup, Suzanne, if we have them? If they're there, that would be, next one, please. The next one, please. Next one, please. Okay, thank you. So I think, as I said in my conclusions, I see in drug discovery that we often have different models. So one of them is, for example, animal models. So we make, in this case, we create these animal models of pain or different pain states, and then we ask the question, we give our molecule and we ask the question, does it improve the pain, and we make objective measures of those endpoints, and we can show, if we can show an effect in that model, we take that as a very positive thing. But that is a model, and it has assumptions with it. We assume the biology in the rat is the same as the biology in the human,
but it isn't necessarily, for example. And I think we also have another model, which is what I call the human brain model. So it's the model where the project teams got together and said, well, anandamide we think is important in pain, so therefore if anandamide is cleared by FAAH, so if we inhibit FAAH, then anandamide will go up and that will have a positive impact. Now, that could be correct, but again, that's a model. And it has certain assumptions associated with it that we can build a model of the complexity of the biology in our brain, that the model's correct. And again, there are assumptions with it. They're just different assumptions to the assumptions of the animal model. And I think one of the prospective values of the QSP modeling exercise is to give us a kind of third modeling approach in the toolbox. So we build a model that's a mathematical model. It includes our understanding of the biology, includes our measured parameters, and again, there are assumptions, absolutely. We assume that we've put in all the relevant biology, we assume that we know it all, we assume that the values are correct. And so there are assumptions there. They're just different assumptions to the one in the animal model and the human brain model. So in my view, one of the clear added values prospectively of QSP is to do that, to give us another element to our toolbox that can allow us to ask questions about our projects, and in that way we can rank them.
Okay, another question from our audience. If you had been a consultant for this company, knowing what you know today, except of course the outcome of the trial, what recommendations would you have provided to them in order to optimize their decision-making process?
So, well, my view prior to the phase 2 was that it was high risk, and that compared to some of the other projects that were running at the time, it was less likely to have a positive outcome. So I understand that people have to, the decisions have to be made, and there are kind of, ultimately this is just the systems pharmacology is a model with assumptions in it. But my view would have been, on the balance of probabilities, and what we'd observed would be that we could have put that on hold and prioritized other targets.
So speaking about targets, someone from our audience would like to know, based on this experience, how would you recommend venturing into new pharmacological targets?
In the endocannabinoid system, I assume. Yeah, so as I said, I think this is a really interesting area, and it's possible that there are lots of useful and interesting drugs targeting elements of it, but I really think it has so many complexities, and it could be so potentially powerful as we're seeing, that we really do need to understand it far better than we do at the moment. And the obvious next sort of branch, as it were, to understand would be the kind of AG element. So I think I would see, if the industry is interested in pursuing drug discovery in this area, to me we must build a better understanding of this kind of endocannabinoid system. And I would start with taking the model that we have and integrating the 2AG axis.
Someone would like to know, was there any reason to believe that the Bial compound was hitting the target-related pharmacology, and if so, was likely overdosed? If not, then how could a QSP approach for FAAH have helped in this case?
So if I understand it correctly, I think they did dose the inhibitor above where we would have seen a kind of maximum pharmacology. Now, I think, as I was sort of trying to say in the end there, that we could have, perhaps some work needs to be done on that molecule to understand what else it's inhibiting apart from FAAH. But with that knowledge, which probably could be generated clinically, and the systems pharmacology model, I think we could have started to ask some questions, some better questions about what was likely to be safe and what was likely to be efficacious.
Another question from our audience, can we introduce human neurophysiology, for example, nerve firing or conduction velocity assays to develop a model linking CB1 to electrophysiology and further constrain the model?
I think that would be nice. Having worked for many years in nerve firing, I think it's one area where our technology needs to be improved. And the kind of understanding of the biology needs to be improved. But I think, yeah, in principle, that would certainly be one of the next steps required.
Someone would like you to comment about the ethical reasons for first understanding the biology of a drug in a drug system before implementing human trials.
Okay, yeah, that's a good question. So I think that's a really important important point. If you know, should we, are we going to continue to look at these kinds of FAAH inhibitors kind of as we've done here? Or do we say we must understand the biology more before we move ahead? I mean, in my view, in my view, we must do that because, well, there's an ethical reason, as you said, but I think there's also a kind of business reason that says we've got to get better at up front figuring out what the right targets are.
Another question from our audience. Where in the drug discovery pipeline does system pharmacology have the greatest impact?
Could you flick up one slide?
Sure.
For me, please, Suzanne.
That right one?
Yeah, so I think I have this vision that that each project ideally should have a systems model associated with it right from the beginning, because early on that would allow you to look at targets across a portfolio and rank them in terms of their likelihood of success. Because I think what we've seen is that even with a relatively simple systems model, you can start to eliminate some ideas early on. So that becomes hugely saving in terms of time, money, and possibly even ethically. However, having said that, and what I've seen as well happen is that model can kind of grow with a project, as it were. So as a project progresses from the preclinical to the clinical, the model can be extended, clinical data can be added, and when clinical data can be added, the kind of confidence level in the model will increase. So I have a vision where, yeah, each project would have a systems model with it, and it would grow with the program right out into phase 3, and then ultimately we have models that will start to integrate with one another. So, for example, if we're looking at different targets in the immune system and we're building models around those kind of models would become integratable at some point.
Looks to be the last question from our audience. What is the value of animal models in pain research?
So I looked at this extensively in the past, and the conclusion I drew was that it depends on how close or otherwise the biology is between the two species. So in some ways, rats and mice and so on, some of their biology is very similar to human, but in other ways it's very different. So, for example, the biology around COX is very different in a rat to what it is in a human. So if you try and translate between effective COX inhibitors between rats and men, they're wildly different. So I think it's something where it's an area where we need to tread very carefully, and yeah, not as this FAAH example shows, just don't assume that a positive in the animal model is a slam dunk in man, because I don't think it is.
looks like we have a few more questions from our audience. Given the complexity of the brain, the permeability, transporter processes, extracellular fluid versus cerebrospinal fluid differences, is a QSP approach for brain targets too difficult until we have a better ability to model brain exposure in the brain, drug exposure in the brain?
So I think if ignoring active transport processes, I think we can do a fairly good job of predicting exposure in the brain based on what I've seen. I think the next horizon, though, is to try and understand what the sort of kinetics and dynamics of that would look like when we factor in, say, PGP or other active transport processes. I think that's less clear at the moment.